Throughout the animal kingdom, the brain separates colour and motion vision into separate pathways. This is thought to reflect a trade-off between distinguishing different wavelengths for colour vision and tracking fine details that are moving quickly for motion vision. Together, these separate pathways are thought to converge to produce an integrated visual perception of form and colour, but they are not completely independent. Growing evidence suggests crosstalk between colour and motion vision pathways with important consequences for overall visual performance. We investigated the integration of colour and motion vision in butterflies, which are remarkable for their exceptionally colourful visual ecologies and their engagement in rapid aerial chases during courtship or when defending territory. We focus on two species of nymphalid butterfly, the monarch butterfly Danaus plexipus and the tropical blue wave butterfly Mycelia cygnaris. These species are markedly different in their wing coloration, but they also diverge in the composition of their retina. Insects have compound eyes formed from multiple facets called ommatidia. Each ommatidium contains a lens which focuses light onto photoreceptors. In butterflies, photoreceptors are arranged in tiers, with different arrangements of photoreceptors sensitive to different colours. Photoreceptor spectral sensitivity can be measured using intracellular electrophysiology, which reveals the change in membrane potential for different wavelengths. In nymphalids, the basic set of photoreceptors are sensitive to UV, blue and green. This arrangement is the case for Mycelia, whilst Danaus contains an elaborated set of photoreceptors extending spectral sensitivity into the red. So we have two species of butterfly with different wing coloration and different photoreceptor sensitivities. We next investigated how colour interacts with motion vision by measuring the spectral sensitivity of motion-sensitive descending neurons, which send information from the brain to coordinate thoracic motor centres. Descending neurons are organised into parallel pathways of neurons sensitive to different types of visual motion. One subset of descending neurons are sensitive to wide-field optic flow. These neurons are involved in stabilization reflexes to maintain steady flight. Other subsets of descending neurons are sensitive to small moving objects and are involved in coordinating goal-directed behaviours such as chasing of conspecifics. So we have two different pathways of descending neurons involved in either stabilization reflexes or goal-directed behaviours. We next investigated the spectral sensitivity of these two pathways using a custom-built multispectral display system. Two mirrors scanned objects of different colour across the butterfly visual field, and we recorded the responses of descending neurons. We found that in both species, wide-field optic flow-sensitive descending neurons showed minimal wavelength dependence, responding to objects across the presented spectrum. We also showed that these broadband responses correspond to those of optomotor reflexes and can be explained by the available photoreceptors in the retina. In contrast, target-selective descending neurons are exquisitely sensitive to spectral wavelength. For example, in monarch butterflies, target-selective descending neurons are maximally sensitive to red objects, with very little response to blue or green. However, in Mycelia, similar neurons are specific for UV and blue objects, with little response to red. To quantify the alignment between target-selective descending neuron spectral sensitivity and wing coloration, we measured the wing reflectance of each species. This shows the fraction of light reflected for each wavelength. We found that for Danaus there was a near-perfect alignment between the target-selective descending neuron spectral sensitivity and wing coloration. We also saw a close alignment for Mycelia, although descending neuron spectral sensitivity was slightly shifted towards longer wavelengths. However, when we take into account the spectral composition of skylight, we find that Mycelia wings actually appear bluer under natural conditions, shifting wing reflectance toward longer wavelengths that align more closely with descending neuron spectral sensitivity. These results show that the butterfly motion vision pathway exhibits task and species-dependent spectral tuning, extending all the way to descending neurons that control the body's movements. This illustrates how evolution shapes neural circuits to match ecological demands and opens up many exciting directions for future work. For instance, where in the butterfly brain is colour and motion vision first combined? Can the spectral sensitivity of descending neurons change across an individual's lifetime? And does scent and olfaction influence the function of these spectrally sensitive motion vision pathways? Butterflies provide an ideal system to answer these questions and ultimately deepen our understanding of sensory motor control.